delete the low-scoring robots and to make randomly modified copies of the surviving robots. And I showed you yesterday, I showed you that this is usually done in a physics engine in simulation. We then attempt to transfer some of those evolved simulated robots from the virtual world to the real world. I showed you some examples of legged robots showing locomotion. I showed you examples of soft robots made from 3D pixels or voxels. I also showed you some examples from object manipulation. And what we're going to do in the first part of today's lecture is look at a relatively recent application of evolutionary robotics to what have now become known as computer-designed organisms. And this is joint work together with my former PhD student Sam Kriegman here at the University of Vermont and our two biology colleagues at Tufts University, Doug Blackiston and Michael Levin. We'll conclude our discussion of computer-designed organisms and evolutionary robotics partway through today. And if we have time at the end, I'll talk a little bit about a related discipline to evolutionary robotics known as machine science. Just as a reminder, please don't hesitate to ask any questions in chat. There's relatively few of us here, so you can also feel free to simply unmute your microphone and ask your question if that's easier. I always prefer these lectures to be as interactive as possible. Okay, so to introduce this concept of computer-designed organisms, obviously we've been building robots from traditional parts for many, many decades now. These parts include metal skeletons, electronics, sensors, motors, batteries, connecting circuitry, and so on. It's interesting to note that 101 years ago, in 1920, a Czech playwright named Karl Čapek wrote a play called Rossum's Universal Robots. It was written in Czech, and that was the very first use of the word robot. This playwright Čapek actually formulated this word in Czech, which is robota, which means something like slave or helper or useful machine. And in that play, the robots were not actually made from metal and electronics, because obviously electronics didn't exist back then. The robots were made from this strange biological material. It wasn't really specified in the play. So it's interesting to note at the birth of robotics 101 years ago, at least the birth in the imagination, in the human imagination of robots, they were built from biological materials, not technological materials. 2 years ago, we published our first paper on computer-designed organisms, which have also become known by their nickname, the Xenobots, and I'll explain why they're called Xenobots in a moment. And these are, again, if you consider them to be so, they are robots that are made from biological materials. Okay, as you can imagine, if you build robots out of biological materials, it attracts a lot of interest. We published our first paper on Xenobots back in January 2020, just before the pandemic started. There was a lot of interest in this work. It was covered by the New York Times in the top right there, CNN in the bottom right, and in a lot of global media outlets. Obviously, you all can probably read the article on the left much better than I can. So something about this technology has seemed to capture the public's imagination. Okay, so why are they called Xenobots? Well, as I mentioned, these are going to be robots that are built from biological materials. And we took those materials from a very particular species of frog known as Xenopus laevis that you see here. So xeno plus robot is Xenobot. But xeno in Greek also means stranger or newcomer or foreigner. So I think both of those interpretations of the word Xenobot are appropriate here. In the very first paper, we drew our materials from two parts of the frog. We borrowed skin cells, and these are skin cells not taken from the adult frog like you see in the cartoon picture here. We actually took these skin cells from very, very early frog embryo, so basically a frog egg. We harvested some skin cells from that frog, early frog embryo, and we also harvested heart muscle tissue. And frog heart muscle tissue, as you can imagine, is when it's inside a frog or even when it's on its own, it's electrically active. And heart muscle cells will increase in volume and decrease in volume spontaneously on their own. I've drawn here a light blue voxel and a red-green voxel to remind you of what we talked about yesterday. Yesterday, I showed you that in soft robotics, sometimes in simulation, we simulate simulated robots made from voxels. And light blue we usually use to represent passive soft material, which is exactly what frog skin tissue is. It can be pushed and pulled by external forces. We're going to represent frog skin tissue as red-green voxels, which you'll see in a moment, which are active. They spontaneously increase and decrease in volume. So we took from our biology colleagues, we took a basic description of how frog heart skin cells work and how frog heart muscle tissues work, and we built that into the simulation of blue voxels and red-green voxels. And then we turned the program over to our evolutionary algorithm. Again, we used the AFPO algorithm, which I walked you through yesterday, age fitness Pareto optimization. AFPO is a particular kind of evolutionary algorithm. And what you see on the left here are 3 of the initially randomly generated Xenobots. Remember, at any one point in time, we have a population of CPPNs, which are generative networks. Each CPPN in the population paints or constructs a 3D structure, which in our case, we're going to interpret as a simulated Xenobot. You can see the basic descriptions of these simulated patches of tissues by looking at these 3 GIFs over here. You can see the Xenobot in the top left, which is mostly made from skin cells, and you can see them being passively pushed and pulled by the red-green cells. Second one down here is made made completely from frog muscle tissue. And the third one is some sort of admixture of the two. You'll notice, if you look carefully, that the red-green voxels are increasing and decreasing in volume, but there's a random phase offset between any pair of red-green voxels. The reason why is we asked our biology colleagues what happens if you rearrange frog heart muscle tissue. Obviously, if frog heart muscle tissue grows in a normal frog and grows into the shape of a frog heart, those heart muscle cells will communicate with one another, and they will learn to synchronize their beat so that they all contract together and they all expand together, which is, of course, what you want for a heart, so that the heart acts like a pump. If you rearrange that tissue, they still increase and decrease in volume, but there's no guarantee that they will synchronize their beat. So we assigned to every red-green voxel in a simulated robot a random phase offset. The fitness function that we're going to use here, we're going to try and evolve these simulated Xenobots to move from left to right as quickly as possible. So in this initial experiment, we wanted to evolve for locomotion. You'll notice that the first 2 Xenobots basically don't travel anywhere, but if you look very, very carefully, you'll notice that this one in the bottom left is actually moving a tiny little bit to the right. So again, intuitively, the evolutionary algorithm would delete these top 2 Xenobots. It would delete the top 2 CPPNs that generate those 2 Xenobots, and it would make a randomly modified copy of this 3rd CPPN that produces that 3rd Xenobot. And when that CPPN is copied, it's going to slightly alter the neural architecture and or the synaptic weights inside that CPPN. And that new child CPPN will generate a new simulated Xenobot, which has slightly different 3D structure and a slightly different distribution of skin and heart muscle tissue. Okay. Any questions so far? So far, so good? Okay. We ran this evolutionary algorithm for several weeks on the supercomputer here in Vermont. Each of these soft-bodied simulations is relatively computationally expensive, so it takes about a couple weeks to run this evolutionary algorithm. And in this very early experiment, this was the most fit Xenobot in the population at the end of the evolutionary algorithm, which, as you can see here, moves from left to right. I'm just going to loop this video a few times because I want to point out a few interesting observations about this particular evolved Xenobot. The first is you'll see that it has kind of these small back legs on the back, and it's got mostly skin tissue on the top. You'll notice that it travels in a more or less straight line from left to right, even though the individual motors, the individual red-green voxels, are acting randomly. They have a random phase offset to one another. If you look at the skin voxels on top, you'll notice that they're kind of absorbing all of the random forces being produced by the heart muscle voxels and kind of smoothing out or combining those forces. So what the evolutionary algorithm has discovered in this particular evolutionary run is a way to build a reliable machine, a reliable robot that reliably moves in one direction, but it builds that machine out of unreliable parts. This is, again, an example of one of the reasons why in my group we like to use evolutionary algorithms, because it often comes up with non-intuitive designs, something that would be difficult or impossible for a human engineer to build. For the roboticists here, think about trying to build a robot from a set of motors, and then we can assume maybe they're rotational motors, and those motors have random phase offsets from one another. You can't reliably assume that they will synchronize or do exactly what you want. How do you get reliable behavior? It's a very difficult thing for a human engineer to do. Okay, so I'm going to talk a little bit about this first experiment, which we published in PNAS back in 2020, and you'll notice I've written down the URL for this experiment as well. I obviously don't have time today to go into all the technical details. You can check out the website to find the actual manuscript, and a bunch of other videos and images, and also some of the source code all of the source code that we used to evolve the simulated Xenobots. Okay, so that's step one of the experiment. We then sent that one evolved Xenobot blueprint to Doug Blackiston, our biology colleague. Doug is a microsurgeon, meaning that Doug looks through a microscope, and under the microscope, he's able to build up almost cell by cell, not quite cell by cell, but close. He basically builds the evolved blueprint. There's a series of videos that show Doug actually building a Xenobot. I'm only going to show you two of those videos today. The first one is this is partway through the construction process. And what you're looking at here is Doug has already removed frog cells from the frog egg or the early embryo. He's placed those individual cells inside a petri dish. So we're looking down through the microscope into the petri dish. And in that petri dish is a small well, a small concavity. And so the cells kind of pool into that concavity. Cells also don't like to be on their own. So you can notice that these cells are kind of moving randomly, and they're generally trying to connect to their neighbors. And when they attach to their neighbors, they're a little bit sticky at this point. They attach to their neighbors and they deform and pull on their neighbors so that they basically recohere or re-come together again. And this is, in essence, we're kind of rebooting multicellularity. We're restarting the developmental process of this one frog. And this is so outside the experience of these early frog cells that they don't develop into a frog. They develop into something else, in this case, just a ball of cells. Doug can then come in with a set of microsurgical instruments and scrape or sculpt the Xenobot to match the blueprint that you see on the left here. So what you're looking at here is we're looking at this Xenobot flipped upside down, and Doug is trying to make these very small 4 legs that you see in the robot on the left. So what Doug is building is actually a 4-legged Xenobot. He's not building the 2-legged Xenobot that I just showed you, but this is the basic process of how they're constructed. As I showed you in the 2-legged robot, there was heart muscle tissue on the lower half of the robot and skin tissue on the upper half. So when Doug is sprinkling cells into the dish, he puts down 1 layer of heart muscle tissue, another layer of skin cells, heart muscle, skin, heart muscle, skin. So basically building it up like a sandwich or a hamburger, depending on the design, and then scraping away unneeded material. So part 1 of the experiment is evolutionary algorithm and simulation, and part 2 is microsurgical construction of one of these evolved Xenobots. And as I explained to you yesterday, the hope is that the physical robot will exhibit the same kind of behavior that we saw in simulation. And it turns out, again, not all of the time, but some of the time, the physically constructed Xenobot, which you're going to see in the bottom part of this video, moves more or less like the evolutionary algorithm predicted that it would. You'll notice that the movement is not exact, and the shape is also not exact. I'll play that for you again. We wanted to guarantee that the movement from left to right of the physical Xenobot was because of the evolved shape and distribution of tissues. So we took that physical Xenobot, which you see in the top, and we flipped it onto its back, which you'll see in a moment. When we flip it on its back, it no longer moves. So it's definitely using the 3D shape that evolution discovered. Okay, just to give you a sense of how big these Xenobots are, you can see one of these Xenobots in this plate here. It's a little less than 1 millimeter across, so they're just visible to the naked eye. Okay, in this first paper, at the end of the paper, we tried out some other ideas, like, for example, putting multiple simulated Xenobots in a dish together. And when you do, I'll ask you just to focus on the top half of this video for a moment. When they do, and if you sprinkle additional pellets, simulated pellets into the simulated Petri dish, you see that they kind of push these pellets around. And because of the complex 3D shape of these Xenobots, from time to time when they bump into one another, they partially interlock and they stay connected for a little bit before pulling apart again. They weren't evolved for this collective behavior. We evolved them for locomotion and then put them together to see what would happen. We did the same thing in the bottom half of this video with the physical Xenobots, and you get some similar behavior. You see that they also kind of attach together a little bit. And if you look very carefully at this video, there's a couple of very small glass beads that are in the dish, and these Xenobot, they're much smaller black dots, and you can see these Xenobots bumping into these beads and moving them around a little bit, which suggested to us that we might be able to evolve Xenobots to perform collective behavior. Here's zooming in on one simulated Xenobot in the top that was evolved to actually push a pellet. And then we found a physical Xenobot, which you're going to see in the bottom half, which seems to be interested in this one pellet. And this Xenobot, again, the physical Xenobot in the bottom half, was only evolved for locomotion. It wasn't evolved for pushing objects. It's important to remember that the Xenobots are built from biological cells, and every individual biological cell is itself a robot. A cell has many, many sensors, many, many motors, and cells have very complex internal decision-making processes. It's possible that the Xenobot that you see in the bottom half is actually sensing and reacting to that pellet, even though we didn't evolve it to do so. Again, this is very different from traditional robots where any one part of a robot, like its metal or its arm or its battery or its sensor, a battery itself is not intelligent, a sensor is not intelligent, a motor is not intelligent. The components that make up traditional robots are not intelligent, but the components that make up biobots are. And this is a very different idea in robotics that's important when we think about trying to build robots from biological materials. Any questions so far? Questions, comments, ideas? No? Okay. Okay. Again, this is a brand new technology. We're not sure what the applications might be for this technology. There is, within robotics, there's a lot of interest in what's known as intelligent drug delivery. That someday we may be able to make very small micro robots that you can swallow or ingest. And those small micro robots may be able to carry a small capsule of medication and intelligently deliver that medication to some particular point inside the body. It seemed to us that if you can make robots out of frog cells, you may, in the not too distant future, be able to make biobots out of human cells. And if you could make them out of cells from the human patient themselves, you could construct a self-bot, something made from your own cells that could work safely inside the human patient's body because that robot would be made from the human patient's own cells. And therefore, that biobot would not trigger the human patient's immune response. So we played around with this idea of evolving a biobot to carry a small yellow packet of medication. This was just sort of like a thought experiment. It's not something we can try in reality quite yet. One other interesting thing about building robots out of biological cells is these robots inherit a lot of the abilities that cells already have. Cells have 3.5 billion years of experience with dealing with all the terrible things or the difficult things that can happen in the real world, like physical injury. So you can see in this case, we're cutting this biobot almost in half. And over a few minutes, that biobot is able to deform its shape and actually seal up this wound. Imagine taking a traditional robot or even your laptop and cutting it almost in half. It is definitely not going to recover. So when we evolve robots or design robots with biological materials, we inherit a lot of functionality for free. And that's kind of an interesting thing to think about, I think. Okay. Again, one of the advantages of evolutionary algorithms is that it doesn't give you back just one design. It gives you back a palette or a Pareto front, which we talked about last time. A Pareto front of diverse designs. In this particular case, we actually ran 100 AFPOs. So we ran 100 independent evolutionary runs, and at the end of each of those 100 AFPO runs, we took the most fit Xenobot, and you can see it here. So again, blue is skin tissue, and red is heart muscle tissue. Somewhere in here, there we go. I hope you can see my cursor. That's the one that I just showed you that we actually built in reality. Turns out that most of these 100 designs, Doug Blackiston, our microsurgeon, was not able to build under the microscope. Some of them are hollow, it's very difficult to build. Others had more complex geometries. So again, if we want to try and transfer robots from simulation to reality, it's good to evolve not just one design, but lots of diverse designs. One of the advantages of evolutionary algorithms over other machine learning methods. I'm going to skip over the second paper, which we published in March of 2021, that focused on collective behavior. You can go and have a look at that paper if you like. I'm going to focus now in the next few slides on the third Xenobot paper, which we published just last month in December. And like the first Xenobots, but walk, those first Xenobots look and act nothing like normal frogs. In this next experiment that I'm going to show you, the Xenobots also perform a behavior that has no analog in nature, and that is what's known as kinematic self-replication. What you're watching here is a group of what you're watching is a group of Xenobots, and instead of sprinkling glass beads or inorganic material into the petri dish, in this case, we're going to sprinkle more frog cells. These are individual skin cells now in this case. And as you can see, depending on the shape of a Xenobot, it will move about and push some of these cells together. And you'll notice these cells start to cohere or bunch together into a pile. And it turns out that if some of those piles have enough cells in them, they will start to grow what are called small cilia. And cilia are very small hairs. An individual cell can have hundreds or thousands of cilia on its surface, so tiny, tiny hairs. And these cilia beat like very small fins or very small fins and exert a very small force, impulse force against the surface of the cell. And if a group of cells, if a Xenobot has enough cilia on its on the surface cells, that's enough for the Xenobot to start swimming. So in all of the videos and the work I'm going to show you now, none of these Xenobots have heart muscle tissue in them. They're 100% frog skin cells. But these skin Xenobots are able to not walk, but swim. You can't see the hairs because they're so small in these videos, but they're able to swim. And these small piles that the other Xenobots make, if they get large enough, those piles grow cilia, and those piles themselves, after about 5 days, start to move. And those moving piles are child Xenobots. The quote-unquote parent Xenobots have replicated, and they've replicated kinematically, meaning based on motion. So all replication or reproduction in nature happens by growth. So trees grow seeds on their outsides, and mammals grow their offspring on their insides. So lots of different strategies in nature, but it's all growth-based reproduction. Here we're looking at a form of replication, reproduction, that is not based on growth, it's based on movement, moving around and collecting external material into a functional copy of yourself. That's kinematic self-replication. And so you can check out the website there, kinematically replicating organisms. KR-ORGS stands for kinematically replicating organisms. Okay. So that's the basic idea. I'm going to show you some of the technical details of this project now, and then we'll switch to talking about machine science. So at the beginning, Doug Blackiston again builds about 9 or a dozen Xenobots and puts them all in a petri dish. That's what you see in panel A here. In panel B, as I just mentioned, Doug sprinkles additional individual frog cells into the dish. And as those 9 parent xenobots move around in the dish over about 24 hours, they push some of these cells into piles. And those piles are labeled with the O's in panel C here. The P stands for parent, and the O stands for offspring. These piles are not moving quite yet. In this case, we then take all of these big piles, all of the piles that are labeled with an O, we take those piles and we put them into a new dish, a new Petri dish in panel D here. And we let them mature for about 5 days. And at the end of 5 days, some of these larger piles, again, if they're large enough, they will grow cilia. If they're too small, they don't grow cilia. Why that happens, we don't know yet. So after about 5 days, these larger piles start moving. And in panel F here, if we sprinkle additional frog cells into this second dish, those offspring will in turn start to push piles into the next generation. And this process can repeat a number of generations. But we notice that after a while, this process halts. So at some point, there aren't enough piles to mature into moving piles. And because there are only a few moving piles, there's not enough of them, and they're not big enough to make new piles. So those new piles can't develop into the next generation, and the process halts. Turns out that this halting process happens pretty quickly. So if Doug makes these spherical or round xenobots, basically what happens is these parents in A make the offspring that you see in D, but those offspring are too small. They don't grow cilia, and the process never makes it to panel F. And with probability alpha, this process halts. So manually built xenobots can only kinematically self-replicate for 1 generation before halting. So in this case, we are now going to bring in an evolutionary algorithm, and we're going to ask the evolutionary algorithm to design the shapes of the progenitor xenobots, which are the ones in panel A here. Progenitor meaning the very first generation, the ancestors, if you like. It's going to try and design the 3D shape of these xenobots. And the fitness of these shapes is not how fast they move or whether they manipulate an object. The fitness is how many rounds of replication these evolved xenobots can accomplish before halting, before the piles are too small and can't move anymore. So in summary, what we're going to try and do is evolve xenobots to delay, to postpone the loss of replicative ability. Okay, so this is a quite different fitness function from anything that's been tried before. So let me explain, and again in a little more detail, how this works. So in panel A here, I'm now representing in these circles here. This is our initial random population. And as always, each one of these circles represents a CPPN. That CPPN can generate a particular shape. So in this case, this particular member of this particular CPPN in this initial random population generates this particular shape. We take that shape and we make 9 copies of that shape in the simulator. 1, 2, 3, 4, 5, 6, 7, 8, 9. We have 9 copies of that particular shape, and we now observe how well this particular shape does at building piles, and how well those piles. So in this particular panel E here, we can see that 9 of these xenobots produced 1 to 3 piles, and it turns out that those piles are big enough to grow cilia. So in the next generation in simulation, those 3 piles start moving. And if we put those 3 piles in a new simulated Petri dish, as you see in panel F here, they start pushing those piles on their own. And in this particular progression here, 3 of these initial children, one of them produced a big enough pile, which became 1 grandchild. Okay. What I just described to you is actually this evolved solution here in pink, which is out here. So let me just back up for a moment. As I mentioned, this particular CPPN here produced this shape. We copied this shape 8 times to produce 9 progenitor simulated xenobots, and those nine simulated xenobots moved around in the dish, and they did not make any piles big enough to make it to the next generation. They made 1 pile that was 74% the size needed to start to grow cilia and to start moving. So the fitness function actually gives a few points here. It gives a little bit of a score for building larger and larger piles. So it turns out that in this particular initial population of random CPPNs, none of them were able to produce piles big enough that started to move. But this particular CPPN produced a pile that was almost big enough. It was 74% of the way there. So it got the highest score. This particular CPPN survived. These CPPNs that produced smaller non-moving piles were deleted. And the evolutionary algorithm made, in this case, 3 copies, randomly modified copies of this one. This CPPN produced a pile that was 69% of the size required to start replicating. This copied and modified CPPN produced piles that were 93% of the size required to start moving. This one, C, this particular CPPN produced this shape down here. It was a spherical Xenobot with a little hole in the middle. And for whatever reason, 9 of these shapes, 1, 2, 3, 4, 5, 6, 7, 8, 9, when those 9 were moving around in the dish, two of those 9 happened to together push some of the simulated cells into a pile that became large enough that it started to move. So these 9 progenitors collectively produced 1 child that was able to move, but that child was never able to make another pile that was large enough to start moving. Okay, so in this case, this particular CPPN got a score of 1.51. It made 1 pile that became a child, and that child produced a pile that was 51% of the size required to produce the next generation. Okay, so the higher the score here, the more likely that CPPN was to survive and produce offspring. And what we're showing here is a particular family. Remember, I talked about families yesterday when I talked about the AFPO algorithm. So it turns out that this individual actually survived in the population for some reason, just by random chance. And it happened to produce some offspring CPPNs that eventually produced this CPPN down here, D. This particular CPPN generates this particular shape. If this shape is copied 8 times to produce 9 versions of this shape, these 9 collectively pushed the cells around and made 3 piles. Those 3 piles are shown in F here. And these 3 piles collectively produced 1 final pile, which you see in panel G here. So these 9 grandparents produced 3 children, which one of them produced 1 grandchild. So you can see by going from B to C to D over this evolutionary process that the evolutionary algorithm has been able to postpone the loss of replicative ability. It was able to produce more generations. It turns out in the results of this paper, we were able to actually evolve, xenobots that replicated 5 generations before the process halted. And when those simulated designs were built in reality, those physical xenobots were able to produce 5 generations of replication. And the shape of that evolved xenobot, you can see here, it's this C shape with a little bit of a cut in the front. And as you'll watch in this video, you can see, not surprisingly, that as these C-shaped xenobots move, they happen to just capture more cells in their mouth. And just by chance, that causes them to create larger piles, which produces more children, which produces more grandchildren, and so on. So here in this setup, shape corresponds to replicative ability, and evolution is able to search this space, searching for xenobots that have higher replicative ability. Okay. Again, in H here, what you're seeing is the results of 100 separate AFPO runs. And I just walked you through one AFPO run is visualized in panel A here. And sorry, these are not 100, they are 7 by 7, 49 different AFPO runs. And you'll notice that most of them are some sort of variation on this C shape. Okay. So I'll show you a video of these simulated xenobots now in action. Here we go. You can get a sense of how the evolutionary algorithm discovered this idea. I'll play this a couple times just so you can see what the evolutionary algorithm, I'll play this video a few times so you can get a sense of how these simulated xenobots work. Okay. So the last part of this paper that I want to talk about is the engineering side. So obviously from a scientific point of view, it's interesting to bring in AI, in this case an evolutionary algorithm, to explore all of the ways that genetically unmodified biological material can be rearranged to produce new kinds of organisms that have never been seen on Earth before. This is obviously a kind of a surprising result in biology. All of us were taught in high school that frog DNA produces frogs, human DNA produces humans, but it turns out that that was an overly simplistic story. DNA does not encode frog or human or jellyfish or what have you. What genes encode is the way to build cells and how those cells interact with their environment, including their neighboring cells. If you alter the initial conditions of an early organism so that there are different cells talking to one another and it's in a different environment, very different from what it ever experienced in nature, it turns out that it will develop into something that looks, in certain circumstances, it will develop into something that looks and acts very different from what you would expect. It's interesting from a biology point of view. I realize everyone here is not a biologist, so I'm going to switch back to the robotics aspect of this work. One of the ideas that's been around in robotics for a long time is how to make self-replicating robots. This is an idea that was actually originally devised by John von Neumann in the 1960s. And John von Neumann actually came up with a theoretical idea. This was never built in reality. Basically developed the theory for self-reproducing self-replicating automata. So he mapped out the conditions under which a machine, a theoretical machine, an automaton, would need in order to produce functional copies of itself. Von Neumann himself thought this would be a useful way to approach robotics if we want to do things on a large scale, because it's very difficult to build millions and millions of robots to collectively do something together. It would take a long time to build those millions of robots. It would be very expensive. It would be better to build one self-replicating robot and have that self-replicating robot build 2 more, which would build 4 more, 8 more, 16, 32, 64, and so on. So with very small financial and technological investment, figuring out how to build one self-replicating machine and finding the resources to build it would be relatively easy to do. Once that gets going, then you have your self-replicating automata. If you can design self-replicating automata that they actually do 2 things, they self-replicate and they do some useful work in the world, you would get what's known as exponential utility. One useful robot does a small amount of useful work. It makes 2 robots which do twice as much useful work. 4 robots may do 4 times as much work. And if you imagined time on a horizontal axis and total utility, the total amount of useful work performed by the robots, that curve of utility would increase exponentially. Okay. That's been an idea and not a reality since the 1960s. And the Xenobots are still not quite there yet, but I would argue they're one step towards making these useful self-replicating automata a reality. We explored this idea in simulation in this paper. In this example, we put self-replicating Xenobots in a particular environment. And again, I'll put this video on loop because I'm going to talk about this video a little bit. There's a lot going on here. So imagine we introduce these Xenobots into an environment where they're capable of doing some useful work. In this case, we imagine that these self-replicating Xenobots in purple have frog cells nearby, which are the green cells. So they have access to the building blocks from which they're going to make new Xenobots. So that's the self-replicating part. But we also put some blue components into the Xenobots environment. And these blue components are meant to represent very small wires. You'll notice that some of these wires are attached to a battery, so you can see yellow current flowing outward from a battery. And if these wires come into contact with one another, we imagine that they're sticky and they attach to one another. So these are not traditional wiring that you would find in a circuit. These are small, flexible, adhesive or sticky wires, and they attach together. And if you watch this video a few times, you'll notice that in the 4 corners are 4 light blue towers. And those light blue towers represent very small light bulbs or light sources. You'll notice that through the collective action of the Xenobots, they cause some of the wires to attach, and some of them close a circuit that flows from the batteries out to the light sources and back again, which turns on the light. So we're assuming that together these Xenobots, the useful work we want them to do is circuit assembly. They're trying to they're trying to put together these parts of a circuit to close the circuit and turn on the light. Obviously, this is in simulation. We're making lots of assumptions here. This could not be done in reality yet, although there is a lot of work on making flexible electronics. There's a lot of work on this for soft robotics, because obviously in soft robotics, the body of the soft robot is going to deform, and it would be useful for obviously the electronics to deform in shape along with the robot. So that's kind of where we got the idea for this. Okay, so given this setup, we can now test this concept of exponential utility. Osamu-san asked the following question. If this is off topic, please skip this question. Do you think there is a need for something like consciousness in evolutionary robotics? I think that we need a sense of agency, a kind of consciousness to control our body. Is it possible that something like consciousness could arise in evolutionary robotics? That's a great question. We can pause for a moment and talk about that, and then come back to exponential utility. One of the aspects of robotics that I've always found interesting, and part of the reason why I got into robotics in the first place, is that robots challenge us. They challenge our preconceptions about what we think about thinking. In the study of AI and robotics, you'll hear terms like agency, which Osamu-san mentioned here, agency, consciousness, decision making, free will. What am I missing? Emotions. All of these concepts, they feel real to us. They feel like we have them. Osama-san says, I think we need a sense of agency. It's something that we need to control our bodies. It feels like that, but from a scientific point of view, whether that's real or true remains to be proved. So I think robots, and especially evolutionary robotics, are a great tool, not just as useful engineering machines, but they're useful scientific tools. They allow us to really explore this issue of what does it mean? What do these concepts actually mean? And in my personal experience of working in this field for 20 years, what these robots have shown me is that we don't have a good idea at all what these terms actually mean. And we have no good way of measuring whether they exist or not. You may feel as if you have agency or consciousness or emotions. You assume that your friends and family, your fellow humans, also have agency and consciousness and emotions. But if I was to ask you what scientific proof you have that other people have agency, free will, consciousness, I would guess that you'd have a hard time answering that question. What is the proof? How do you know that your fellow humans have these abilities? Question gets even more complicated when you talk about animals. Do animals have agency? Do they have consciousness? How much? You might say that a dog or a cat has consciousness. Whether you think an insect does, or a bacteria or a virus, again, it's very hard to know where to draw the line. It gets even harder when we talk about robots. You can create a robot that acts happy, but whether it is happy is a very different question, and a very difficult question to answer. So again, Osama-san, I think this is a great question, and it's not one that I have an answer with, and I don't think a lot of other people have an answer with as well. It may be that someday, maybe the next generation of roboticists like yourselves will figure out how to build robots to test whether robots have consciousness or can have agency, and whether we do as well. So again, this is one of the great things about robotics. They're useful from an engineering point of view. They're interesting from a biology point of view, especially in the case of the Xenobots. They're also interesting philosophical tools. I hope that's a good response to your question. Thank you for that. Great question. Okay, back to self-replicating Xenobots. We know that they self-replicate. What we're interested in here is, do they have exponential utility? So the way that we tested this, as you see in panel A, is we took this evolved shape, this sort of C shape or Pac-Man shaped Xenobots. We put them in a dish with other frog cells, and the black components, remember, are parts of circuitry. And we counted over about 20 seconds in the simulator how many light bulbs were turned on. In this particular run, these 9, I think there's 9, 1, 2, 3, 4, 5, 6, 7, 8, 9 of these Pac-Man, they managed in 20 seconds to connect 3 batteries to 1, 2, 3 light sources. And this light source over here remained off. So this particular group of 9 Xenobots did some useful work. They turned on 3 light bulbs. At the end of those 20 seconds, we took all the piles that had been made. So you can see these irregular piles here. And it turns out we took all those piles and all of the parents, and we counted up how many parents and piles there were, and we took that group as a whole and split it in half. In this little cartoon example here, we put half into one new dish with 2 batteries and 4 unlit light sources. We took the other half of piles and parents and put it into a 3rd Petri dish down here, which also had 2 batteries and 4 unlit light sources and some other pieces of wire. So we've got 2 new dishes that contain 2 new partially built circuits. And we're going to see whether the parents and children together turn on any of these light bulbs. If they do, we keep growing and splitting. Sorry, I forgot to mention, in the Petri dish B and Petri dish C, we sprinkle more frog cells into the dish. The individual cells, we didn't draw in A, B, and C for clarity, but they're there. So assuming that in any one dish, they manage to self-replicate and turn on at least 1 light source, we split that group and put them into 2 new Petri dishes, split again and split again and split again. If in a given dish, they don't manage to produce piles that mature and start moving, we stop and we stop this process. We don't split and continue this process on. What that produces is this figure that you see in D here, that you can see here, and I'm going to explain what this figure represents. In the very center of the circle, you'll see there's a small red dot. That red dot corresponds to A. That dot is 1 Petri dish, and that Petri dish is colored red because in that dish, the swarm turned on at least 1 light source. So you'll notice, again, you got to look very carefully here. Coming out of that red dot are 2 edges, which represent Petri dish B and C. I guess it's a little too small to see, and I don't think I can zoom in like this. There are 2 lines that are coming out of that dot, and if both of those Petri dishes within each of those 2 Petri dishes, if at least 1 light source is turned on, there's 2 new Petri dishes that are constructed from there, and so on and so forth. It's a little difficult to see in this picture, but you'll notice that some of these branches end early and they're gray. So a gray edge represents a Petri dish in which there weren't enough Xenobots to turn on any of the 4 lights, and the process halted. So you'll see some gray mixed in with red, but you'll see that there were enough dishes in which useful work was done. At least 1 light source was turned on, and enough self-replication occurred that there was enough to put into 2 new dishes that this process continued. And if you think about the concentric rings inside this picture, starting from the center and moving outward, we're moving outward in time. So t = 0 is at the center of this dish. The next t = 1. The next round of replication is the next loop, is the next circle out. Time = 2, time = 3, time = 4, and so on. And if we unroll that time along the horizontal axis here, and we measure over time, over rounds of replication or splits of the simulation, the total number of lights that were turned on, the number of lights that are being turned on in these growing number of Petri dishes is indeed increasing exponentially, proving that at least in this simulation setup, using these evolved shapes, exponential utility is possible. The dashed line here corresponds to a control experiment where we assume that there was N robot arms. Remember the Google arm farm? Google's arm farm that I showed you at the beginning of yesterday's lecture. So imagine we have a series of arms, and in parallel, they are all trying to connect small pieces of wire to turn on these lights. Over time, these N arms will turn on N lights, and at the next time step, those N arms will turn on another set of N lights, which means there's a total of 2N lights turned on. The next time step, 3N lights have been turned on, and so on, which is a linear increase in the number of lights that are turned on. So what's represented, what we're really trying to represent in panel E here is the more usefulness of self-replicating robots compared to traditional robots. And in my personal opinion, this is the future of robotics. Not building robots that build circuits or build cars or build laptops. It's building robots that build robots and cars or robots and laptops and so on. How to do this in reality and how to do it well is, again, something that is beyond our current ability. But for the young roboticists that are here today, I challenge you to see if you can figure out how to do this in reality. There's another question here. Says, sorry, it's a little bit long. We consider a new hand or finger that is combined with soft robotics with mechanical hand or finger. When would such a model in artificial general intelligence genetic algorithm is utilized? Oh, if a genetic algorithm is used to evolve the surface of the mechanical hand or the finger of the robot for grasping instead of muscle and skin, what should we do to switch from simulation to real robot as our first step? Okay, I think I understand this question. So if you evolve the surface of a soft robot hand or finger, how do you start in on sim to real? That's a very good question. The first thing I would suggest is to see if you can evolve in simulation soft surfaces. There are a lot of soft-bodied simulators that are available, that are out there. I showed you VoxCAD, which is the one that we use. There are many others that you can use. One of the things you want to look for is, one of the things you want to try and do is, first of all, just evolve in simulation to see if you can discover something like the jamming gripper or something that exploits the softness of the hand for gripping, and not worry about whether that will transfer from simulation to reality. Once you've accomplished that first step, the next thing to think about is what features of those evolved designs are unlikely to transfer to reality. And some candidates are non-linear behaviors. So trying not to get into too much detail, but if you think about the material properties of soft materials, you can think about dynamic and static stiffness. Sorry, not explaining that well. You want to make sure that there are not high nonlinearities. So sticking and slipping in the contact modeling between surfaces. You want to make sure that things move very slowly, and that a slight change in the deformation, the slight change in the soft fingers or the soft hands, don't suddenly cause a complete change in the way that the hand and the object contact one another. So you want to make sure that there's a linear relationship. A small deformation in the hand causes a small change in the forces acting on the object that you're trying to grip. That's a difficult thing to do by hand. So I suggest in the second step, you make an evolutionary algorithm where one axis, one of the fitness objectives is grasping the object. So how well does a given hand grasp the object? The other axis is the amount of nonlinearity in the interaction. So you can then evolve things that maximize gripping success and minimize nonlinearities in the grip. Those kinds of, and you that'll give you a Pareto front again, where you can select some of the evolved soft designs that exploit the softness of hands and fingers, but do so in a linear manner. Those things, linear relationships, are more likely to transfer from simulation to reality, because if this is the simulated design and this is the physical design, there's obviously going to be differences in the physical structure of the simulated design and the physical design. That's okay, because if you've evolved something that's linear, slight changes in the 3D shape or the material properties of the hand or the fingers are only going to affect the ability to grasp by a small amount. I hope that makes sense. If it doesn't, please feel free to ask your question again. There's another question here. As the theme on the current page, is it an evolution of robots to increase more and more lights? Is the object to increase the number of lights turned on? Professor Hayashi says thanks. Okay. So sorry, back to Ryogo. I'm sorry if I'm mispronouncing your name. Ryogo's question. On this page here, we are not evolving robots. We have evolved robots already. We have these C-shaped Xenobots, which you see in panel A. Evolution is already finished. What we wanted to do is ask the question of whether these evolved robots are capable of exponential utility, meaning over time, more of them produce more useful work. And it turns out that the answer is yes. Okay, so you asked, is the object to turn as many lights on? Yes, that's kind of the object or the goal, but the real goal was to try and prove exponential utility. This is more of a theoretical exercise. As I mentioned, this isn't something that we can achieve with real Xenobots or real robots yet, but I think this is something that could be possible in the next few years. Raji-san asks the following question. Thank you for this interesting topic. My question is about the lifetime of the Xenobots and how we keep feeding them with enough energy to stay alive. That's a great question. Remember that the Xenobots are, in these experiments, they're only made from skin cells. There's no neurons, so there's no nervous tissue. And they also have no digestive system. So these Xenobots have no way to eat. They can't take in any nutrients. I didn't talk about the energy source for these Xenobots. Excuse me a moment. The Xenobots are powered by an internal power source, which is egg yolk. Remember that I mentioned that we take these skin cells from very, very early frog embryo, which is basically a frog egg. So the cells inside that egg have absorbed or pulled inside of themselves egg yolk, which is obvious. Yolk is evolved to be energy rich. And the cells that make up the Xenobot gradually digest. Each cell inside a Xenobot digests the egg yolk over the period of about 7 or 10 days. At the end of 7 or 10 days, one of these moving Xenobots will slowly stop moving, and then it just rots away like any other dead animal or plant does. So they're not able to keep going. They're not able to extend their lifetime by eating. Um, another question we're interested in the years to come in this research is, can we build new kinds of cell types into these xenobots? Can we include new kinds of organs like digestive organs? Could we create more complex xenobots that are able to eat and digest and extract energy from their environment? Not quite there yet. Okay. That's all I wanted to say about computer designed organisms. So I think this is a good point to pause. I just wanted to confirm with Professor Hayashi that we have about 20 minutes left, 15, 20 minutes. Is that right? You can just
Yes,
type.
yes.
Okay. Okay. So I'm going to spend the last few minutes talking about something slightly different, but I think will also be of interest to this community, which is this idea of machine science. So this is an idea about basically automating the scientific process. So as all of you may remember from high school, I assume this is taught in high school in Japan, you formulate a hypothesis, you come up with an idea about how nature works. You then think up an experiment, and that experiment, when carried out, will generate data, and that data will either support or disprove your hypothesis. You come up with a new hypothesis, new experiment, new data, new hypothesis, new experiment, more new data, and you go around and around and around, which in English we call the scientific cycle, or the scientific process. It's interesting to think about trying to use robots to automate this process. And I'm going to show you some older work about how this works. This is taken from a paper by myself and Hod Lipson, which we published in Science back in 2006. So if you Google Bongard Lipson Science 2006, you can find this paper and have a look yourself. In this case, it's the robot itself that is going to act as the scientist. And the hypothesis that this robot is going to try, the hypothesis that this robot is going to generate is how it's constructed. So we're going to assume that this robot doesn't know anything about its own body. It doesn't know how it's put together. We're going to give this robot a little bit of initial information. We're going to tell this robot that it's made up of these 9 parts, but we're not going to tell it how these 9 parts are put together. So the robot can generate a hypothesis, a guess about its own body by attaching these 9 pieces together in arbitrary ways. And then once the robot has that hypothesis, I think I'm put together in this way, the robot can generate an experiment. It can send current to its motors and move and get back the sensory information, which is the data. So the hypothesis is how the robot is put together. The experiment is an action that the robot performs. That experiment generates sensor motor data, and that sensor motor data is going to be used by the robot to prove or disprove its current hypothesis about how it's put together. Okay, that's the summary. Let me show you a little bit more of how this works. I'm going to walk you through an experiment. I'm going to walk you through one run of this process. At the very beginning, the robot has no hypothesis, doesn't know how its 9 parts are put together. The robot is actually going to start not with a hypothesis, but with a random experiment. It's going to move. In this case, the robot is going to rotate. It's going to rotate motor 1 and motor 5 down, and it's going to rotate the other 6 motors up. So it's performing a random action. Imagine you're in a black box, and in front of you are 8 levers, which correspond to the 8 motors. So this robot knows it has 9 body parts and 8 motors. You're in this box, you have these 8 levers, you pull levers 1 and 5 down, and you push the other 6 levers up, and you sense that you tilt to the right. So this robot has just two proprioceptive sensors. Those proprioceptive sensors tell the robot how much the robot is tilted left and right. And the second sensor tells it how much the robot is tilted forward and back. So imagine again, you are the robot. You've pushed up 1 and 5 and you feel yourself rotate. You've pulled down 1 and 5 and you feel yourself tilt to the right. That's the experiment you performed. It should suggest a hypothesis in this case. What is that hypothesis? What is it? It should give you a hint about, generally speaking, how your body is organized. If you have an idea, if you think you know, go ahead and type it into chat. If you've pushed motors 1 and 5 down, or the body parts attached to motors 1 and 5, you've rotated those body parts down and you've tilted to the right, what does that tell you? What hint does that give you about your own body? Any ideas? If motors 1 and 5 go down, they're probably on your left side because they went down and you tilted to the right. So what you're going to see in this next video is the robot had just performed a random experiment, and it's going to generate a whole bunch of hypotheses. And what you're watching is one hypothesis after another. In essence, the robot is saying to itself, what happens if the 9 parts are put together like this? What happens if the 9 parts are put together like this? What happens if the 9 parts are put together like this? And for each of those hypotheses, each of those virtual robots, it performs an experiment. It rotates motors 1 and 5 in each of those designs down, and it rotates all the other 6 motors up. And if you look carefully, you'll notice that all of the green cube is tilting forward. And that's my fault. I actually recorded this video incorrectly. You need to rotate your view. This green box is actually always tilting to the right. So what the robot is doing in this case is evolving virtual robots, like I showed you today and yesterday. And the fitness of each virtual robot is when that virtual robot rotates motors 1 and 5 down, it generates the same proprioceptive sensation that it got from reality. So it's evolving accurate self-models. It's trying to evolve hypotheses about itself that match its physical experience. So the physical robot did something and got back a sensory result, and it then says, it hypothesizes, how am I put together? What would explain the action and sensory repercussion that I got? You'll notice with that single action that it performed, this action here, it turns out that there are many hypotheses that are possible. All of these hypotheses are correct. They all explain the experiment. They all explain the experience that the robot got. So the robot doesn't have enough data yet to converge on one hypothesis, the correct description of how it's put together. So the robot goes back and performs a second experiment. In this case, it's going to rotate one of the motors on the right-hand side down, and all 7 other motors it rotates up. Play this a couple times so you can see this. So it's performing a second action, and now the main body is tilting a little bit to the left. So this is experiment number 2. And so it now has 2 pieces of data. I did action 1 and got proprioceptive information back. I performed action 2 and I got this new proprioceptive information back. So it's got more experiments and more data. And the robot is now going to continue evolving virtual robots. I'll put this on loop again. It's going to continue to evolve experiments. And for each one, evolve hypotheses. For each one of these virtual robots, it evaluates that virtual robot twice. The first time, it rotates motors 1 and 5 down. And then the second time, it takes that virtual robot and rotates motor 6 down. That gives 2 pieces of virtual proprioceptive information, and it has to match those against the 2 pieces of physical proprioceptive information that it has. And a fitter robot, a better robot, is one in which both pairs of data match up. The 2 pieces of simulated sensor data match the 2 pieces of real data. There's a closer match. It turns out that there become fewer and fewer and fewer hypotheses that are able to explain both experiments. And if you watch carefully on this video, you'll see some of these bad hypotheses are deleted from the population. And at this particular point in the evolutionary algorithm, which is evolving populations of simulated robots, it evolves a good description of the robot's own body right now. You'll see right at the end of that sequence, it converges on the correct description. The robot discovers through this evolutionary process that it's made of 4 legs, and those 4 legs are arranged around the main body. You'll notice that for a split second, it actually thinks it has 3 legs now, and then continues evolution and discovers the 4 legs. So this is sort of a snapshot of a robot scientist. It's doing what human scientists do. They're collecting more and more data, and they're in essence evolving. They're evolving hypotheses. The hypotheses are changing all the time, but it gets harder and harder to come up with crazy or weird hypotheses. Eventually, the set of hypotheses that explain the growing amount of data gets smaller and smaller and smaller, which forces you to converge on a correct hypothesis, a correct description of what's going on out there in the world. This is something that the scientific community does as a group, but this scientific process can also be done by a robot about itself. What I haven't told you yet is why you would bother doing this. From a robotics point of view, this can be very useful in the field of model-based control. This is something you may have seen in class. Model-based control, as the name implies, is that the robot uses a model to test out control policies before trying out that control policy on the real robot. Model-based control. Okay. In robotics, however, we often write down the model ourselves by hand, or we build a model of the robot in simulation, and that takes a lot of effort. It can take months or years to create a very accurate robot model. What you're watching here is the robot evolving a model of itself. Sylvain, I see your question. I'll come back to it just in a moment. Let me just finish this part. In this case, now that the robot has generated its own model, we didn't have to write down the math or program a simulation of the robot. The robot came up with a model of itself, which you see here, and it's now going to perform model-based control. Here is a control policy for the model, and when we run that control policy on the model, we get what we want, which is the robot to move from the left side of the table to the right side of the table. So in this experiment here, there are actually 2 evolutionary algorithms. One is evolving accurate models of the robot. The second evolutionary algorithm is evolving controllers using the model, doing model-based control. So this was sort of an advance in robotics that was interesting from a robotics point of view. but it's also interesting from this larger view of machine science. How do we automate the generation of hypotheses and experiments and data? Okay, I'll pause there. Sylvain has a question. What do you think of explainable AI as a way to better understand how policy and structure of robots are automatically selected using machine learning, and even theorize those choices after? That's a good question. I think this could lead to a very interesting use of AI as science exploration and not maximization of efficiency for one objective. Sylvain, I agree with you absolutely. One of the problems with more traditional model-based control or traditional robotics is it's often not, explainable AI. You can train a neural network, you can train a deep network to act as a model of the robot, but it's not very explainable. And of course, if you train a neural policy, that neural policy is also not that explainable. So as you mentioned here, one of the things I think that's nice about evolutionary robotics is that it's, as you mentioned, it's less black box. Like everything in science and engineering, there's always a trade-off. So the advantage of evolutionary algorithms is that they are white box, or at least gray box. They're a little bit more visible, as you can see on the screen right now. But in evolutionary algorithms, you also cannot guarantee convergence to an optimal solution. There are other robotics and AI techniques that can converge, that can provide formal guarantees of convergence to an optimal solution. And in many robotics applications, that's the most important thing. However, again, looking at AI in general, there is a question, which is how strict is this trade-off between black boxness and safety guarantees or optimality guarantees? Can we develop new robotics and new AI that is explainable, as you mentioned here, that is XAI, and can also provide formal guarantees of optimal convergence to an optimal solution? No one has a good answer to that question yet. And I think that's a great question for future roboticists to try and answer. We're at 8:18, so I think maybe instead of introducing new material, this might be a good place for me to stop for today. I wanted to thank all of you again for your attention. 90 minutes is a long time to sit still and listen to a lecture. I appreciate your attention and your great questions. And I'd like to thank Professor Hayashi for the very kind invitation to come and speak with you all. Thank you very much. Arigato gozaimasu.
Thank you very much. We are around a lot today, thanks. But we have to study much more to understand your model. So I had a great time with you. Thank you for your continued support.
Thank you so much. And I wish all of the students the best of luck with their studies and future work.
Thank you very much. Thank you.
Bye-bye, everyone. Bye-bye.
これで、終わりますけども、もし、いろいろ、if you have any comments, so actually directly to Professor Josh, he's a very busy man. So actually if you have the question and the comment, so actually to Miss Apilati or Hayashi Lab sent to email, right? Not directly to Professor Josh. Anyway, one question. So actually to our laboratory student or laboratory send an email your question, comment. これで終わります。あとその情報セミナーで登録等々している人は書類を出してください。はい、じゃあ今日はこれで終わります。どうも、ありがとうございました。